the PhD process, and later we'll then have a panel discussion where we'll be trying to explore certain things around PhD, for example, issues related to career issues, career and research areas. And so I just want to thank you guys very much for joining us. I think everybody on board so far knows who I am. I'm Herin Menya, student at Sheffield Hallam University. I'm doing my PhD third year, doing my PhD in mathematics education. Most of you are aware of that, so I will not really get into that. I think we're going to have our first presentation from the first, not really PhD student, because he already has his PhD, so you need to be asking him what business he has with PhD anyway after doing PhD, because a number of you have indicated to me that you thought after PhD no more studying. But anyway, we'll have the first presentation. He is a student at Sheffield University, student, quote unquote, and he's going to be presenting on something around climate change. So let's have our first presenter make his presentation. We will hold on to the questions until the last session. Now, as you listen to all the presenters, we'll have them in a queue. As you listen to all the presenters, I want you to write down, as you listen to them, what you think are the key characteristics for a PhD person. So as you listen to them, what comes out to you as you listen to them as they make statements about their research? What are some of the qualities that you think PhD people have in common? Well, they all may look crazy, so if you think that's the case, just write that down, and you'll write that in your chart, and we'll be projecting and looking at some of the things that you have written, and then we'll be asking them to respond to some of those qualities and add value to that discussion. So without further ado, I think I will ask our first presenter to make his presentation. Thank you, and please stay on and
University
listen.
of Sheffield. And I already finished my PhD 2 years back, but I like research. So I'm continuing my study till now, and hopefully for quite some time in future. So today I'm going to talk about mountains, because I work in mountains, and they are not only a very beautiful place to live, but they are also home for magnificent different kind of flora and fauna. And when you go to a mountain and you started walking from the lower elevation or altitude towards a little bit higher, there are several things which got changed along your movement. That can be different atmospheric pressures, solar radiation, temperature, precipitation, as well as different seasons. And all this combined to create a topography diversity. That means all the places in across mountain elevation gradient are very, very different from each other. And that creates unique habitat for several animal and plants. That's why mountains are such a big, highly biodiverse system. And in mountains, I mainly study this small cute animal called pika, P-I-K-A. There are several species you can find around the world, but I study only one species called Royalist pika in Himalaya, and they are relative of rabbits and hares. And because this animal is so cute, they inspired people to make Pokemon and Pikachu cartoons in Japan. And here, before moving further, we should be able to see a video about American pikas. So they are making calls. So pikas can make burrows in some cases in Tibetan plateaus and Ataram areas. Also, they always use rock crevices as their home, because certain pika species like Himalayan pika species or pika which lives in American Rocky Mountains can't make burrows, so they use these rock crevices as their home. Why they are important? Why people should care about them? Because they are very important prey species for several animals and birds, and because they are rabbits, that's why they don't get these winter hibernation or winter sleep during around probably November till next February, because they're active. So they are the only prey for these, or food for these particular animals and birds which feed on other animals. They make burrows, so their activity on soils influence several soil geochemical cycles. For example, you might have known or have heard of nitrogen cycles as well as phosphorus cycles. So their burrowing activity influence those cycles. And they are herbivores, that means they eat plants, and this grazing activity of eating plants influence alpine plant communities. But then there is a big problem with pikas, because due to change in global temperature, they get heat stress. That means these animals are adapted to high alpine regions, which is very cold. So now their entire body is very thick fur coated. When the moment due to high temperature during the day, they get heat stress. That means they can't have the sweating to get rid of the heat, and they die. Problem during winter is a little different. They like snow because snow acts as a blanket for them, and their habitat or their burrows become very, very warm, so they can live there quite nicely. But due to climate change, the snow thickness are very, very less now, so that expose this animal for the cold stress. And finally, they are herbivores. They eat different kind of foods, but due to change in precipitation and temperature, their favorable food resources are going to be changed or already changes certain areas. That means they are not getting proper nutrition, and these three things combine together to make them vulnerable towards climate change. So I'm studying them in Himalaya, where you can see the red arrow. It's called Uttarakhand. This area is quite famous for its scenic beauty. So this is a Hindu temple for god Shiva. This is the highest Shiva temple in India, and these all other photographs depicts different seasons, certain vulture which is very endemic to that particular region of Himalaya, and flowers. When I worked there, so I also got to see these wonderful snow-capped mountains. And when I'm saying mountain, that means these mountains are more than 7,000 meters in height and always under snow during throughout the year. These are some other animals like Himalayan thar, which are similar to other Ethiopian or other African mountain ungulates. There are Himalayan mouse deer, brown bears, and Himalayan monad, which is the state bird of that particular area where I work. Sorry, there is yeah. So these are the predators like raptors, weasels, martens. These are the animals which eats on pikas. So if something happened to pikas, it actually can influence entire food chain, which can have negative impact on each of these animals. So when I go to field, I generally do trekking. So I have backpack and I go to field to collect data, which I record from my PhD and scientific observations. So I start working from around 2600 meters and I work till 4700 meters in altitude in mountain. Mainly I work in summer, but depending on my research questions, for example, if I need to know what they're doing during the winter or how snow thickness influence their numbers, I also have to do or conduct studies during the winter months, when I go and measure snow and try to note down if pikas are active or inactive, if there is other animals I can see there or not. So once I go to my field, I have permanent plots, and then I use my binoculars to see those animals, their numbers, if it's a male or female. Sometimes you can say, most of the time you can't, and I note down which kind of habitat they live. Okay. Which kind of food they eat, as well as I put very small thermometers called data loggers. Those are like a small coin, so I put them inside their habitat, as well as on the trees. I get those temperature data, and then I compare them with my other observations. But apart from this field observation, I also collect genetic samples, because now we can address several climate change related questions using DNA. For that, I go to my field, I select certain plots, and I observe where pikas are, and I collect their fecal samples. So I know fecal samples, the moment you got to know about fecal samples, probably some of you are thinking like, oh no, again, not fecal sample. But then fecal samples probably give us wonderful information, because you can extract DNA, not only DNA of the animal it belongs to, but also DNA which it eats. For example, for pikas, it's plants. So I can actually know through the DNA samples which I can extract from these particular fecal matters that which plant it actually ate. And now when I analyze my data to know how vulnerable or how threatened they will be due to climate change, I can have multiple aspects of their ecology, so which I can trace back using not only their numbers as well as their food plants number, how it got changed, how that change can influence pikas number. And then I make DNAs, and then DNA helps us to make estimation about populations, movements, everything. And finally, all this information we put together to make different distribution map, which we provide to the government officials for the further conservation implications. For example, we did this mapping using all the information which I get from my field observation, and we found that around 40 to 52% areas which are suitable for this species now will be lost in 2070. Therefore, we can demarcate certain areas, observe pikas as model system to understand if climate change, if it's happening, which area where pikas is going to respond first, which area they might respond little later. But then, these are the areas which I highlighted might be affected. But apart from climate change in Himalaya, there are mountains in Kenya, and there are several wonderful species which are very endemic. That means they can be only found in Africa or in Kenya. For example, there are small mammals, shrew as well as rats. They only adapted to these high alpine regions. They are the animals which can respond very negatively towards climate change, because change in snow, vegetation can also affect these animals' numbers significantly. So if you think which species you can study to understand climate change in Kenya, you just need to know what questions you are asking, why you think that my animal might be vulnerable or might be get threatened because of climate change, and you select several protocols are there. You select the animal based on those protocols. That okay, these are the characteristics which might make an animal vulnerable or threatened towards climate change. But apart from the research, you got to travel when you do research. So I was born and brought up in Calcutta, which is eastern part of India. But throughout my PhD as well as now, I got to travel in different parts of US, Europe, as well as China, Japan, for conferences, for fellowships. So not only you do research, which is field-based or sometimes can be lab-based, computer-based, but it also allows you to travel around the world, get a very different idea about how people live in those countries, as well as their culture. So now I will, we are going to take the questions later. So kindly note down your questions and get back to us at the end if you have any queries. Thank you.
This evening I'm going to talk about impacts of global financial crisis on the stock volatility of some Nigerian banks. When we talk of global financial crisis, what really happened? We know when we talk of market or financial system is made up of three entities. The entities are the financial market, whereby financial activities or goods are traded. We also have the intermediary, which is the banks, and then the agents, which are the individuals or company or corporations that go to this market to trade. In 2007 to 2009, the problem began in America, where they were having the problem from the mortgage investments, and then the banks, then the market, and there are a lot of problems in the financial sector America. And as the saying goes, when America coughs, the whole of the world is affected or catch colds. The same thing is happening at the level of the financial market. Now, as I've been introduced, I'm Maruf Rahim, Sheffield Alam University. I'm presenting that. Now, how did this global financial crisis happen? What we want to look at, my main focus is to discuss how the 2007 to 2009 global financial crisis, and as a result of those crises, there were reforms. Reforms are kinds of the adjustment in the system that were introduced by the policymakers like Central Bank or SEC, what we call Security and Exchange Commission, in charge of regulating the affairs in the market. They brought reforms. We want to look at how did these two activities impacted on the Nigerian banks, affect the Nigerian banks. Look at the pictures of the popular banks in Nigeria. We have about 10 there. Now, we move on to a brief account of what really went wrong. How did it start? The origin of financial markets. Like I've hinted at the initial stage, we had failures of corporate governance. Household savings were becoming too little because people were panicking because the banks were having the picture of collapse. There was excessive leverage in financial institutions, meaning that there were so many risks when people go to the banks, or they want to withdraw their money, they couldn't get enough of what they requested for, and which is happening across the African countries as well. There were what we call housing bubbles and what have you, as a result of which the financial crisis started in America. So it now led to recession, which the entire global market felt effect. Why is it that the effect of America is affecting other markets? Just for instance, in an household, when you have a father, the mother, when the father catch colds, the mother feels the effects because they are interdependent, they are interrelated. The children, the member of households, the house is in cold periods because they are not in good mood because the father is catching cold. Exactly, when you have a market upon which other market depends, when there is a problem in that market, the entire dependence of that market have the problem. Now, we move on to see what were the implications of on the market. First and foremost, look at what we can see there is what we call series. The blue and the red. The blue was showing one of the big investors in the American market from June 2003 to 2007. You can see that series seems to be uniform in the way it spreads. But come to 2007 July to 2012, you see that one, there's a spike, is the one that appears in red. You can see the uniformity is no more there. At the point, you have a very high spikes along one aspect of the series, and some other aspect are little. It means that at those areas where the spikes are so minimal, it means the market is, let's say, relatively behaving in the same way. Things were normal. But at that point, things started getting uncomfortable to the market. It means the implication is that what happened today in the market cannot be used to predict what will likely happen tomorrow. And that's the implication of such thing. Now, so that series really shows that when there is a problem in the market, there's what we call destabilization, disorganization. So things are not normal. The way the mother cooks in the house, she won't be able to cook again, because the delicacy she has time to cook, because she has to take care of the father, she won't be able to have the mind to do that. Now, look at the effects on the global market. Other markets apart from American market, you have the labor in the Asian market, we have the SGG, we have the TED, and so on. See those markets at a point in time, July 2007, as it happened in America. So you see that those markets at their point of coolness, they started having the headache, and that was why you have the drop till the point of ending of 2008, close to 2009. So the same thing applies to the market. When there is any disturbance, when there's any problem, then the market starts falling. What will happen? There's a risk which we call volatility. Now volatility now is a measure of how the market is moving up and down. When there is excessive stress, just like elastic, when you stretch it so much, it gets to a point of elasticity, and then it breaks at a point. What happened? Look at the picture under that. You have the investors are those people who go to the market. They started losing money, people crying. People were going out of the business. They carried their bags. Instead of going to the market to buy goods, I can't buy it again. Let me carry my money back home. Then what happened? Those market monitors, those market regulators, they are losing jobs. People cannot buy, people cannot sell. So the unemployment increased. Then, on Nigeria experience, because that's my main target, in the first instance, there was no significant impact at the first until the second effect. When did it happen? It started in 2007 in America. But from Nigeria experience, we did not have this experience until mid-2008. That was when the second effect was being felt. And what happened? Because our local policymaker, they have a very strong monetary policy with which to tighten the market, not to allow the external force to come in. Just like when you have a house, when there is a crack in the house, lizard comes in. But when there is no crack, no rat can come in, can penetrate. At that point, there was they tightened the policies. They didn't allow the market to be affected. Unfortunately, there was a limit to which you can hold a falling wall. So before it falls on you. So we started feeling it. Until what happened? Look at what happened. One of the banks. People got to the bank. They got disappointed because the banks were closed. What really happened? The banks and other financial institutions started losing the values of their investments. They go to market, instead of raising funds, they are losing money. And banks don't generate money. They keep deposit of individuals. When they go there, the intention of raising more money. Unfortunately, instead of having profit, they were making losses. So the money in their domain could not be enough to satisfy the needs of the customers. Near collapse of total confidence in the banking system because the total governance there is disorganized. The bank's inability to maintain the capital adequacy. You go to market, want to withdraw £10,000, you were not able to get it. You get only £5,000, of which out of your money that you kept there. Now, then why do we have to target banks? The intention behind targeting banks is that in an economy, bank is so strategic that they play intermediation role between the market and the individuals. Because as an investor that you want to go to the market to buy shares in a company, you don't go directly to buy in the stock market. You have to go to the bank, you pay the money to the bank, and the bank will transfer to the company, then they will share some stake, which you call shares of these banks, of this company, corporation. Then banks play a very significant role as intermediaries. And also that because when there is any crisis in the bank, when there's a collapse of banks, then the entire economy will collapse. That is why it is strategic for us to look at what happened in the past, how to mitigate the effect in the near future, so that our banks will be protected. When we protect our bank, we protect the market, we protect the entire economy. And that is why this research is very important to us. Now, the data coverage and presentation, we have period from 2004 to 2014. This entire period, we have the period of bank consolidation. There were activities that came up. That consolidation was like a reform prior to the global financial crisis in the Nigeria banks. Then during financial crisis 2007-2009, then we have the post-reform 2009 to 2010. Then, now, what is important is that one of the banks, that is what you call price series. You see that from before 2008, the bank, after the first reform that we call consolidation, the bank's stock prices was rising at a point till 2008, mid-2008, that's to show the implication of the crisis in the market on the trading activities on that bank. Then you have a sharp drop. And sharp drop, you could now see that that drop continued like that, struggling to rise. Unfortunately, could not rise, come down, go up, come down, until the end of the study. It shows that when there is too much pressure on the market, then the crisis continues to linger for a long time before you can gain it back. The same way you play your life, when you have played your life, when the first immune system in your body as a result of your carelessness, you play it round, there's no amount of treatment that will be given to you that will make your body to restore back to the normal. Something is lost. So that's why it's important for us to watch everything that we do. There's a series. At the point you have heavy spike is when there is heavy crisis. The same thing with another bank, showing that the crisis also affected the bank almost the same time it affected that first bank. Yeah. Now you have the spike as well. Now I would like you to look at the summary result we've got. When we did measure with fitted model, now this result is showing persistent rate in volatility. I've told you earlier on that volatility is a measure of performances or risk within the markets. When the risk is below 0.5 within a period, it shows that when the persistent level of that risk is below 0.5 or within 0.5, the market is doing well. It means that the investment in that market, in that particular stock, is very nice. And look at what we have there. You see that the first bank there has the highest above 0.5, which shows that if there's any problem with that particular bank, investor is likely to have some risk. It's not too bad. Other ones were minimal. Until July 2007. Look at 2007, we have 1.04, 1.01, 1.06, except the last bank, which has less than 0.1, which is less than 1. It means that all those banks with persistent level above 1 shows that if there's any shock, both the liability of the customers and the banks will be lost into it. Exactly, that was what happened within those period of financial crisis. Before then, you have August 24 to 26. It means that the first reform, the impact of reform, make the bank investment to be what? Stabilized. Unfortunately, when the crisis came, there was problem. After that, we have the second reform, 2009 to 2014. Like you saw in the series, that though the bank were struggling to rise, unfortunately, they could not meet up to the level at which they started from. So you could see that out of the 4 banks, only the 3 other ones were able to bring their level of risk below 1, while the first bank could not regain that its stands again. So now, source of risk to investment, we have historic source, we have the current source. It means that if there have been problem yesterday, the problem today, you pull it together to be able to know how that market or bank is doing in the market. That's a measure. Like I mentioned, when it's less than 0.5, when it's between 0.5 and 0.1, it means that the risk will persist for some time. It means that if there is any shock, the shock might be positive or negative shock in this case. When there is negative shock, it shows that people will continue to lose money for a long time. When there is positive shock into the market, to the system, it shows that people will continue to make gain. And they will make gain to the level of being beyond the necessity, what we call overvaluing the stock. So that's it. Now, to conclude, we can see from the explanation the 4 banks were impacted significantly at varying degrees, and also that the persistency volatility during the financial crisis was very high compared to other periods. And that is the implication of my presentation, of my research for now. Thanks for listening.
Lifetime PhD research. I want to, in this next 10 minutes, I'll look at my watch as well, take us through 4 steps. First, what is a PhD? Second, how you already have the skills to do one. Third, what mine is about and what I learnt along the way. And fourth, look at the toolbox you need for research writing and research thinking. Right, now from where you're standing or sitting, a PhD probably looks like the end of a long educational journey. You've done primary school, secondary school, you'll go on to do a BA or a BSc, an MA, an MSc, and then get onto a PhD. And you think that that's the end of the road. But from where I'm sitting, I feel as though it's the beginning, which you probably find quite worrying now. It's the start of a career as a qualified, certified researcher. But of course it's sort of an end, because what I've completed, and I'm going to hand in my PhD and defend it in January or February next year, I've completed and defended an argument, because that's what a PhD is. It's writing an argument. And the argument in my field, in humanities of history and English literature, is 80,000 words, which is about 250 pages of A4 paper, 1.5 spaced. So it's not impossible. It's challenging, but it's not impossible. And I think a good way to think of it is think of writing about 30 or 35 essays of 3,000 words. So if you can do 30 essays, you can do a PhD. But the trick is how you tie it together. And I think you already have those tools. You've probably got sort of the seeds of the tools, but you've certainly got them. So the second point of this talk, in which I'm not going to talk about what you know, which is what the previous 2 speakers have mostly focused on, but a bit what the first speakers sort of touched on towards the end, which is the how you go about doing it. And I think you've already taken the step, because I don't think the big step or the big task is actually getting the ideas, but then like our colleague here, I haven't had to go up mountains in the snow. I've sat in libraries. So from my point of view, it's not actually reading through lots and lots of 19th century periodicals that's been the big task, going to Manchester or going to London. It's actually how to present an argument to somebody when you're actually dealing with very, very complex things. I deal with complex plots, complex laws and complex ideas about changing notions of property. So I've got these 3 complexities, and I've got to unravel them and give them to somebody who knows something about the area but not all of it. So I don't want to speak down to them and sort of make it too obvious and too descriptive, but on the other hand I don't want to make it too abstract. And I imagine that's a problem we all have as PhD researchers, knowing how to pitch it. And you have it as an A-level student or an undergraduate student. And we all have it when your teacher writes in the column, so what? You know, because what you've done is you've sort of described something, but you haven't actually used that finding, that information you found, in an argument. And we all have, we have that feedback from reviewers when we write for periodicals, and we've already got a doctorate. We have so what in the column. You've got to make all your findings, everything you discover, everything you put in your essay, you've got to make it work to push your argument forward. So I think that's one of the things that we all share as PhD or A-level students or undergraduates. And that's actually the difficult task, making a clear argument that's well backed up. And I've found that I've got better at doing that over the past 3 years. And I think it probably applies to you whatever field you're in, the humanities, social sciences, or hard sciences. And the task is to get across complex stuff really clearly. Yeah? Framework something that initially seems dense and messy, and you analyse it and you have to sort of represent it to a way that's more palatable, that's more comprehensible. So perhaps this is the moment, the third step, at which I say what I've done. Now my research is about novels in the 19th century and what I call the inheritance plot. And the question I ask is, why is the primary structure of so many novels in the mid-19th century that of the inheritance plot? And I analyse a group of novels to show that they're structured by this sort of plot. That is, people who pass on property as a gift, normally when they're dead, to someone else, or in anticipation of their death. And I ask, why at the moment when, according to Victorian and modern critics, market relations come to the fore, people start to relate to each other with contracts, legal contracts and cash, rather than with custom and tradition as the main ways of relating to other people, of conducting social and economic relationships. Why, in the main cultural form of the 19th century, the novel, there isn't much representation of the market. It's all about gift relationships and inheritance. And to answer this question, I took a set of, well, quite a big set of novels, but I eventually pared it down to 4 novels by a writer called Wilkie Collins, who was a friend of Dickens, and focused on these novels to make it manageable. As you start your PhD and you're going to do 101 things, and then you're halfway through, you're doing 50 things, final year you're doing 20 things, and you end up doing 3 things. And that's, I think probably everyone's nodding in the room here. Now the work you're most familiar with, I imagine, will be Dickens' Great Expectations, and we know that's about someone who expects property, he gets property, it's not from where he thinks it is, and there's constant discussion about portable property in it. And we find out it comes from Australia and it's dodgy money, and it raises lots of very interesting questions. So I think that's probably the novel you'll know, and it's got a complex plot. Now Wilkie Collins' best novels, if you have read any of him, are The Moonstone and The Woman in White. And these novels, along with Dickens, were some of the best sellers of the mid-19th century throughout the English-speaking world, and they were translated also into French, German, Russian and Dutch, and went the world over. Why were they so popular? Yeah? Over the past 30 years, lots of researchers have written about Collins' novels and linking them to other popular writers, connecting them to the world beyond the novel, assessing the degree to which the world which they show relates to other accounts of mid-19th century Britain, found, for example, in the newspapers, in magazines, in Parliament, parliamentary debate. All of these researchers say how complex his novels are, how complex his plots are, but no one has yet explained why his plots are so complex, nor have they noticed that each is structured in the same manner. And in fact, what happens at the beginning of each of them is you have a will, the terms of a will, what someone's going to live to whom, and then you have, of course, that this doesn't happen. The will doesn't work. The legal document doesn't sort of give the money to the daughter, it goes to a dodgy uncle, or it's left to somebody else, but the money goes down in value. Various things happen so that the path predicted is impeded and doesn't happen. And the third thing they all have in common, and they all happen in the same order, is that we discovered that the reason for this impediment, the reason for the money not going where we expected, was because there's been a previous dispute about a will in the past. Yeah, so they've got this repertoire of 3 elements, which nobody's noticed. And I think, well, okay, why is this happening? And I try and explain why Collins plots are complex, and yet how this doesn't stop us reading them, but it actually creates a page turner. And to demonstrate this, I thought, okay, how important is inheritance in the mid-19th century novel? Let's have a look at the magazines these fictions were first published in, and the newspapers and parliamentary debate. Who was talking about inheritance? And in fact, nobody's really discussing inheritance, but they're all talking about a different sort of property transfer, which is the sort of transfer that happens if you set up a joint stock company and have limited liability. That is, if money is transferred from person to person to make more money, in the way we have now with corporations. Previous speaker talked about stocks and shares. Stocks and shares didn't exist in the form we know them today until a series of acts were passed in Britain from 1840 to 1860 called the Company Acts. And most people in Britain, the government, landowners, other people, were very hostile towards the passing of these acts because it meant that people would become irresponsible with their investments. In fact, that's what we see in the world today when people invest their money in a company in Bangladesh where sort of women are burned alive because they're not, they're not protected and they haven't got enough they're not looked after. And yet most people who have their money in stocks and shares don't realise that that's where their money's going, or to some sort of dreadful diamond mine in South Africa or wherever. What they care about is getting money back from the money they're invested, not what their money's done. So there are these huge debates in the 1850s and 60s at the time that these novels are written, which are all about money and responsibility. And they're all about what responsibilities go with owning money. Yeah? Who should money go to? How should things be distributed? And the way in which these debates are played out in the mid-19th century in Britain is in the novels and in narratives, the way in which banks explain themselves and financial journalists explain how money works, how companies work, is using short story forms and novel forms. So we have things like the autobiography of a shilling, the autobiography of a stock and share, the autobiography of a joint stock company. So there's a very close relationship between fiction and finance in the mid-19th century. And one of the things I think is most extraordinary is people don't know what a stock or share is. Yeah? Because if you ask most people today, they don't know what it is. And effectively, what it is what you own is no longer a concrete object. It's actually you own the future profits that something could make. On the other hand, it might not make that. So there's lots of debates because property becomes very, very abstract in the 19th century. And in my thesis, I demonstrate how this concern about very abstract sorts of property, finance capital, which is really, really new, and all the debates that are going on about it, are played out and explored in the novel form. And I demonstrate that this is what shapes the novel. So, in order to deal with these complex things, an abstract notion of property, new legislation, complex plots, I have to use exactly the same toolbox as you do at A-level or at an undergraduate level. I have to think about my audience. What are they bringing to the table? What do they know? What do they need to know? In how much detail? And at what point of my arguments? How much repetition do I have to give? How do I have to signpost it? How often do I have to say, I'll go into this later in more detail, just believe me for now, I'll show you the evidence for this later. And how much do I need to signpost back, as was discussed in chapter 3, section 2? What pace do I give this information at? How much knowledge do I assume? And you're probably already doing this in the sort of essays you're writing at school or as undergraduates. So basically, I'm doing the same thing, but I just have to write sort of 35 essays relatively quickly and make sure they all hang together, which is probably the trick of it. So really, what we're talking about, 80,000 words. 80,000 words is 8,000 words introduction, 5,000 words conclusion. That's 13, which leaves probably 5 chapters then. 5 chapters of about 13,000 words. And 13,000 words is about 5 sections of about 2,500 words. So that's how psychologically I have managed to get through the last 3 years by thinking in terms of 35 short essays. Okay, I hope that's been useful and thrown some light onto what it is to do.
Research with AstraZeneca, a pharmaceutical company here in Sweden. And then I'm aspiring to do a PhD for, considering the fact that I myself am also an aspirant. And I was informed by hearing that secondary school students and high school students or undergraduate students will be joining in this session. So I'll mostly be targeting them. And obviously sharing my experiences so far, so it's going to be brought to non-specific spectrum of what I've been doing as far as research is concerned and part of my career as a scientist. So the first thing when you aspire to do a PhD is to determine the motivation. What's the motivating factor? Is it because you want to get a PhD title, or because the society respects people with a PhD? From my perspective, I think my motivation will be to contribute to change, to knowledge, to research and innovation, and to inspire others. And in order to achieve that, we need some effort. It's not impossible. It's not something that is abstract, but we need to make a move. We need to take a step towards that goal. And just like the first presenter talked about rabbits being so inspiring and so nice, coincidentally, I also had a rabbit and some carrots. It could also be a source of inspiration. So what I want all those, or the younger ones aspiring, or maybe thinking of a PhD career, or maybe aspiring to be a PhD, is that PhD is attainable, but it's not given. You need to achieve it. And just like Julia said, having a PhD is not the end, it's just the beginning. For me, my journey towards getting a PhD has not been very smooth, as you can see on the picture. It's been characterized by ups and downs, but then all we need to do is to focus. So a little bit on my background. This is not supposed to be a CV, but it's just supposed to highlight some of the things I've done in the past. I had my bachelor in medical laboratory science in 2010 in Cameroon, where I come from. And in 2011, I did a short program in project planning, management and evaluation. And then I had the opportunity to work with the tuberculosis regional reference laboratory in one of the regions in Cameroon in 2012. 2013, I moved over to the National Public Health Laboratory, where I worked as a scientist and project monitor on tuberculosis projects. In 2015, I had a Commonwealth scholarship, which permitted me to do my MSc in molecular medicine and specializing in cancer option. And presently, as I said, graduate researcher. And in 2017, the journey will continue. And obviously, there will be more ups and downs, but then I have to stay focused. So far, one of the things with research is that you have to publish like what you do. And I happen to have co-authored this. I happen to have been a co-author of this study, which we did in 2015. Basically, it's based on this equipment that was developed by a Japanese company. And it's supposed to be used in diagnosing tuberculosis, and it's supposed to have a better performance than existing computer techniques as well as microscopy. So what we had to do was simply to recruit patients, collect sputum from them, use this equipment to analyze the samples, and then use the results from this equipment to compare with other methods that were currently in use. So this is just like what we did. We collected the samples from the patients in hospitals, hospital samples, and then we extracted DNA from it, and then we analyzed the samples, and then it gave us some sort of a readout which you could see visually at the level of detection. So this is already, we completed this study before I move over to the UK and to continue with the write-up, and it's already due to be published. So it's something that is possible. It's possible to do sciences, it's possible to engage in research, it's possible to also share the work you do in research with others through publications. So what we did was we analyzed the samples with the equipment, and then we compared it with the gold standard, which is like the main, the ACE detection technique for tuberculosis. And then we also compared it with a molecular technique, and then we also compared it with microscopy. So that was it for that study. And I'll be sharing the results when the paper is published. And then for my MSc, I was interested in cancer because it's a huge problem back home that is not currently being addressed, because all focus is on tropical diseases. But then there is a problem with targeting cancer, or with addressing the problem of cancer. And the problem is that current cancer treatments are like non-specific, and it has lots of side effects, one of which is hair loss, because treatments are kind of toxic and it affects hair follicles. So people on such treatment always have like hair loss, and then for most women they tend to just shave because it's more convenient. And then there's also an issue of bone marrow suppression. The drugs are non-specific. It doesn't target just the cancer, but it also targets normal cells of the body. And it also targets the bone marrow. And when it targets the bone marrow, since most of the cells in the body are produced by the bone marrow, most patients will suffer from maybe low white blood cell counts, low red cell counts, related effects. And then there are also other side effects like cardiotoxicity, which simply means that it has a negative impact on heart function. So as a solution, we're thinking around understanding cancer, biology of cancer, and how we can develop new treatment methods which are more tolerant and has less side effects. So I was looking at these tiny peptides which are naturally occurring, and they have been found to occur in bees. Actually, peptides are like protein subunits, building blocks of proteins, and scientifically we tend to call them amino acids. So a peptide is just a short sequence of a few amino acids. And I was also trying to combine these tiny peptides to nucleic acids. Nucleic acids are like DNA or RNA or derivatives of DNA and RNA. And to see how these nucleic acids can or could be delivered to cancer cells specifically. And how do these peptides work? Well, getting a little bit into biology. What you see here, like it's a membrane. What you see in blue and yellow and blue is the cell membrane or the cell barrier. And what you see here with the red head and the green tail is just a designation of our peptide, and do is that they assemble into tubes that look a little bit like pasta tubes. And the peptides are attached to the cell membrane according to what is known so far by an electrostatic interaction. Electrostatic simply means that positive charges are binding to negative charges, because cancer cells differ from normal human cells in the sense that they express negatively charged surface molecules on their cells compared to normal cells, which are usually neutral. So our peptide is positively charged, and it's able to recognize the negative charge through interaction, and it binds to the surface of the cancer cells, and it's able to disrupt the cell membrane of the cancer cells, and the cancer cell ends up dying. And it's kind of specific because the peptide is able to recognize just the cancer cell and not the human cell. So we see that if we use our peptide to treat the patient suffering from cancer, it's going to kill just cancer cells and not normal cells, and hence we have small side effects. And this other structure in blue is just a summary of our peptide when you look at the chemistry of it. What is below is just the characteristics of the peptide. Currently, I'm working on lungs of patients suffering from chronic, what we call, it's a pulmonary disease or a lung disease, and it's common amongst people who smoke. It's simply one of the symptoms of the disease is shortness of breath. So people tend to suffocate. They have difficulties in breathing, and you have to give them drugs that will improve on their breathing, or maybe you can always supplement your breathing with oxygen or some other means. But then what I'm looking at are another group of tiny peptides which are produced by epithelial cells. Epithelial cells, or simply cells that line, is kind of altered in patients suffering from pulmonary obstructive pulmonary disease. What I'm doing now is studying the law of production of these peptides in normal individuals, and then I'm comparing in patients, and then trying to understand what's happening as far as production of this peptide is concerned between normal individuals and patients. And that's it for what I'm doing. I'm hoping that after this, or while doing this, it's going to give me some ample time to make a good decision, or to have a better option, or a good option of what I want to do during my PhD. The journey continues. So thanks a lot. I was able to
Thank you, Shady. I think now we are on to our last presenter from Kenya. She's going to make a very brief presentation, and then we're just going to have a wrap-up discussion for 10 minutes, and we'll call it a day.
Hi, my name is Faith. I'm from Kenya. I'm an architect, and my PhD is on sustainable design. But even before I talk about that, because I'll not spend too much time talking about my PhD, half because I just started, so I'm 3 months into the PhD now, so I think we started with the most senior PhD researcher, and then ended with the most junior researcher. So I'm still trying to get a footing on the PhD, but maybe just because we are talking to probably from high school, and I think maybe some guys in campus, it's more to explain how I got here. So of course then I was in high school like you at some point, and